Hello everyone. Today we are going to continue the chapter number 5, Molecular Basis of Inheritance, Theory Part 7. So in today's class specifically we are going to deal with what is transcription unit. We have touched upon it in previous class, but today we are going to go deep into that. And then what is the process of transcription, what is the difference of transcription between eukaryote and prokaryote, and different type of RNA we are going to talk about in today's class. Disclaimer. So what are the learning objective of today's class? First, we will understand the structure and component of a transcription unit. So in previous class we have studied like what actually a transcription unit is, promoter, then the structural gene, and then the terminator. Today we are going to deal with it in more details. Then we will define what is genes and their relationship to this transcription units. Then we are going to talk about different RNAs, that is what is mRNA, what is tRNA, and what is rRNA, which RNA polymerase make these help in transcribing these RNAs. We're going to talk about everything. Then we will explain the 3 stages of transcription. Like what are the different stages of transcription? Initiation, how would the transcription initiate, then how it elongate, and at last how it get terminated. Then we will identify the different key enzymes and factors that are involved in transcription process. So these are going to be our today's learning objectives. So at the end of the class we are going to answer all of these questions. So today's index will be like this. First we're going to talk about transcription unit and the gene, which is NCERT section 5.5.2. Then we are going to talk about the next section, that is 5.5.3, and the topic is about types of RNA and process of transcription. Then we are going to give a gist of today's class in the form of summary. Then there will be some NCERT based questions. Then they are followed by keynotes. And then we end the slides with interesting questions. And we know that after slides get over, we do 5 DPP, that is the daily practice problems. So now let's start with today's lecture. First is transcription unit and genes. So if someone says us, define what is gene, what we are going to say? We are going to say that gene is the functional unit of inheritance. So gene is responsible for this what? For gene is the content which is going to transfer to the progeny. Gene is something that is going to transfer the inheritance to the upcoming progeny. So this is the functional unit of inheritance. It is located where? It is located on DNA. And if we talk about gene in terms of DNA sequence, then it became very difficult to define what is a gene. So here what we got to know about gene, that this is the functional unit of inheritance. Okay, this is something going to take part in inheritance, and it is located on where? It is located on DNA. Example, DNA sequence coding for proteins, tRNA or rRNA. So the DNA sequence that is going to code for protein is what? Is a gene. The tRNA, the DNA sequence that is going to make tRNA is a gene. DNA sequence that is going to make rRNA is a gene. So what is a gene? A functional unit of inheritance which is located on DNA. So if we look at the structure, what we are seeing, that there is a cell. Okay, in cell there is chromosome, and in chromosome, what is a chromosome? So chromosome is condensation of chromatin. And okay, and what is a chromatin? It is the nucleoprotein fiber. Nucleoprotein fiber has what? DNA molecule and proteins like histone proteins, non-histone proteins. So in this chromatin, what we have? A sequence of DNA. Okay, that will be the functional unit of inheritance, and that's what we call a gene. Now, what actually more deeply we will understand what a gene in upcoming slides, like how we decide that this sequence, DNA of sequence, is a gene. Before that, let's understand what are regulatory genes. Okay, so if we talk about regulatory genes, so these are the promoter and regulatory sequences are loosely defined as regulatory genes. So the DNA sequence, which we say this is a promoter, okay, the sequence of promoter and any regulatory sequence, we are saying that this is gene. These sequences do not code for any RNA or protein. These sequences control the expression of structural gene. So what they are saying about that, what if we gonna talk about what are the regulatory genes, so the promoter and any regulatory sequences, we define it as regulatory genes. These sequences do not code for any RNA or any protein, but what they are doing, they are controlling the expression of the structural gene. In previous class, we have studied that what is a transcription unit. So there was a promoter, then there was the structural gene, and then there was the terminator. And we have seen that how this promoter and terminator is flanking the structural gene and responsible for controlling the expression of those structural gene. Now, after knowing this, there are a few terms that we have to know first. Then we will come with the whole concept. Now, what is cistron? A segment of the DNA coding for a single polypeptide. So, a DNA segment which is going to code for a single polypeptide is what we call a cistron. If we talk about the cistron, what is a cistron? Example, the structural gene in transcription unit is often referred to as cistron. So the structural gene in the transcription unit is often defined as cistron. Why? Because it is going to code for a polypeptide. Now, if we talk about the type of the cistron, so first it is monocistronic genes. The gene contains only one cistron. As the name suggests, monocistronic gene, mono means one. So the gene contains only one cistron, and monocistronic genes code for only one protein product. Definitely, because it is having only one cistron, so it is going to code for only one protein product. Example, so the eukaryotes. Okay, the eukaryotes have monocistronic genes only. So this is the difference between a monocistronic and polycistronic gene. Here you just see monocistronic, because when I will tell you about polycistronic, then we will see the second diagram. You see the first diagram, what it is. So 5' to 3', it is a strand. Okay, this will be like promoter region and this will be like terminator region, because we know that the structural gene is flanked by these two. So what is this red color? This is the cistron. This is the gene which is going to code for the polypeptide. So here in monocistronic, we can see there is only one cistron which is going to code for the polypeptide, and one cistron is going to code for how many polypeptide? Only single polypeptide. Now, if we talk about polycistronic gene, what is a polycistronic gene? The gene which contains multiple cistron. So a gene that contains multiple cistron is what we call a polycistronic gene. Now, because it contains multiple cistron, it is going to code for multiple protein products. Example, prokaryotes have polycistronic genes only. So if you guys can see, what is this? They have shown a this, that there is a promoter, there is a terminator, but in between there is cistron 1, cistron 2, cistron 3. Now what RNA polymerase will do, it will convert, transcribe it into mRNA, and now this mRNA is going to code for 3 different polypeptides. Okay, so this is a difference between a monocistronic and a polycistronic gene. Now after this, guys, understand that right now we are understanding different terms. Okay, later we are going to merge all this term and understand what actually a transcription is all about. So till now what we have studied? We have studied what is monocistronic, what is polycistronic, okay, what is a gene, a definition of a gene, and now we are understanding what is exon and introns. So the eukaryotes have split genes. First we have studied what the eukaryotes have only monocistronic genes, means they are having a cistron that is going to code, they have only going to have one cistron that is going to code for only one polypeptide. Along with that, the eukaryotes have also what? Have split genes. In eukaryotes, the monocistronic genes have interrupted coding sequences. Now what does this interrupted coding sequence means? The genes are actually interrupted by exons and introns. Means that gene is going to have 2 regions of exons and introns. Now we have to understand what is this exons and what is this introns. So here you guys can see what is this. So they have showing this is an mRNA, okay, and in mRNA there is exon, there is intron, then there is again an exon, and then there is again intron and exon. So what is this exon and what is this intron? Exon is the coding region that is going to translate into protein, whereas this intron is not the coding region. This has to be removed from the mRNA to make it to the mature mRNA. That is what this figure is showing. You guys can see in this figure what is showing, that there is a mRNA, okay, 5' gap it is having, then this is 3', and all this AA polyadenylation we are going to study in today's class only, so don't get tense about this. So this is an mRNA. What this mRNA has? Exon, then one intron, then again exon, then one intron, then exon. But you guys can see through this process of splicing, they have removed this intron, and now an mRNA, a mature mRNA has formed, which has only exon region, which has only coding region that is going to translate into protein. So this is a tableau form where we can understand this exon and intron more accurately, more precisely. So if we talk about exons, what is exons? As I said, the coding sequences or express sequences are defined as exon. So what is an exon? Is exon the coding sequences, okay, which is going to express, is what we call exon. And now if we talk about intron, the exons are interrupted by these introns, which are what? Which are non-coding sequences, means they are not going to translate into any protein, they are not going to get expressed. Exon, if we talk about again, so the sequences that appear in the mature processed RNA and are translated, means the sequence that is going to be in mature mRNA, okay, and which is going to translate into protein is what we call exon. Whereas what is intron? Intron or intervening sequences do not appear in mature or processed RNA. Because these are non-coding sequences, they are intervening the sequences, they cannot appear in mature or processed RNA. That's why we have to remove it from the sequence to make it to a mature mRNA. So this is the difference between intron and exon. Now we will understand about prokaryotic. We have seen that our eukaryotic has what? They have intervening, means they have this exon and intron region both. But if we talk about prokaryote, they are going to have continuous gene only. Prokaryote have continuous gene segment only. They don't have any intervening sequences like introns. They don't have introns. So here you guys can see. And what we have talked about, prokaryotes are what? They are polycistronic, means they are going to have multiple cistron that is going to code for multiple polypeptides. So here you guys can see. So they have talked about prokaryotic gene. So this is the regulatory region for transcription initiation. It is going to be promoter and all those things. Then this is the coding region, the structural gene, which is going to code for the protein. Then there will be transcriptional termination signal with here we're going to have a terminator and like that. And a prokaryotic gene is continuous, it looks like this. But if we talk about eukaryotic gene, then we can see here that there is a regulatory region for transcription initiation. Then there will be coding region, which we call exon. Then there will be this non-coding intervening sequences, what we call intron. Then there will be like some coding region. Then there will be like termination signal like this. So we can here see that prokaryotic genes are continuous genes, but whereas the eukaryotic genes have what? This eukaryotic genes going to have a lot of intervention. Now, what we have studied, like what is gene, what is cistron, what is monocistron, what is polycistron, then what is exon, what is intron. From that we got to know the different concepts of like what is a gene, and then if how prokaryotic have polycistronic but continuous gene, whereas the eukaryote have only monocistronic gene but intervening gene. But they do have intervening genes in between, what we call introns. Now we have to understand like what are the different types of RNA and the process of transcription. So if we talk about the types of RNA, first is the messenger RNA. Okay, what is a messenger RNA? The mRNA which provides template for what? For protein synthesis. Then this tRNA, what we call transfer RNA, they carry amino acid as amino acyl tRNA. So on this what happens, so on this tRNA, that is transfer RNA, the corresponding amino acid goes and attach, and this whole component what we call amino acyl tRNA. And so when this amino acid go and attach to its corresponding tRNA, there is a catalyst which is going to make this process happens, which attach this amino acid and tRNA, and what we as whole call amino acyl tRNA, and the enzyme which is a which catalyze this process is amino acyl tRNA synthetase. Okay, and what is the structure called? Amino acyl tRNA. What they do, they read genetic code for amino acid. They read that genetic code for amino acid. So here in the figure you guys can see that this is what? This is the mRNA, this is the template for protein synthesis. Then this is the transfer RNA, this is the transfer RNA, and this is the amino acid that is attached to its corresponding tRNA, and this whole structure what we call amino acyl tRNA. And this is something, this whole structure is what we call amino acyl tRNA, and this tRNA is going to do what? This tRNA is going to bring this amino acid at ribosome. Because why? Because we know that ribosome is what? Ribosome is the site of protein synthesis. So now this amino acid will come at ribosome, and then they will read this genetic code and they will make the protein. Okay, the amino acid, and then this we know that the chain of amino acid what we call protein. And this is what? This is the this is the messenger RNA. This provide what? This provide template for protein synthesis. Then we talk about the third RNA, that is the rRNA, which is the ribosomal RNA. It plays dual role in translation. It acts as a structural and catalytic role both. If we talk about the structural role, forms large and small unit of ribosome. So ribosome is always made up of two subunit. Okay, one larger and one smaller subunit. So we know prokaryotic has what kind of ribosome? 7ts. Eukaryotic has what kind of ribosome? That is 8ts. And now the 7ts and 8ts have two subunit. One is large, one is small. So that is going to form large and small subunit of ribosome. And if we talk about the catalytic role, what is the catalytic role in that? It helps in the formation of the peptide bonds. We know that proteins are attached with peptide bonds, so it helping in making peptide bonds. So these are the three kinds of RNA. Now after learning every term like what is gene, what is polycistronic, what is monocistronic, exon, intron, different type of RNA, now it's time to understand that what is actually the process of transcription. So first we are going to understand that what is the process of transcription in prokaryotic transcription. So before starting that, what we have studied that prokaryotes have what? They have continuous gene, means they don't have any intervention gene. They are polycistronic, that they are going to have different cistron and they're going to make for polypeptides, different multiple polypeptides. And what we have studied about prokaryotes? First polycistronic, then that they are continuous and they don't have any this. So they don't have any introns in it. Okay. So the key enzyme which is responsible for the transcription of prokaryote, they are DNA dependent RNA polymerase. So a single type of DNA dependent RNA polymerase synthesize all type of RNA, that is mRNA, tRNA and rRNA. So what we are saying here, the first thing that the key enzyme that is responsible for this prokaryotic transcription is that this DNA, it is DNA dependent RNA polymerase. So a single type of DNA dependent RNA polymerase will synthesize all types of RNA, whether it is mRNA, tRNA, rRNA. Every RNA is going to get synthesized by DNA dependent RNA polymerase only. Polymerizes RNA depending on the DNA template. So why we are calling it DNA dependent RNA polymerase? Because we know that it is going to make RNA is going to polymerize RNA depending on what? Depending on DNA template only. That's why we said that DNA dependent RNA polymerase. So this is, you guys can see, what is this? This is a DNA, double-stranded DNA. And in yesterday's class, we have studied everything like, what is the coding strand, what is the template strand and all those things. I won't repeat it again. But here we guys can see that this is what? This is a double-stranded, double helix structure of DNA. And this will be what? This will be the template sequence. Okay, template strand. Whereas this will be coding strand. But let's see, this is the template strand and we guys can see that RNA polymerase is doing what? Using this DNA as template, it is polymerizing the RNA. So here we guys can see that. Let's go ahead. Now what is the nature of prokaryotic transcription? It is coupling, means this transcription and translation. What is transcription? Like DNA transcribe into RNA. Okay. And what is translation? That RNA is going to code for protein. So both transcription and translation are coupled in prokaryotes. Means it happened together in prokaryotes. But whereas if we will see in eukaryotes, okay, we will talk about eukaryotes later. First see prokaryotes only. So in prokaryote, transcription and translation are what? They are coupled, means they take place simultaneously. And why it is happening? Because we know that prokaryotes don't have any nucleus, they don't have any well defined nucleus. That's the reason this transcription and translation are taking place together simultaneously. So here we can see that what is this? This is a DNA template. Okay, this is the direction of transcription. This is the RNA polymerase. So what RNA polymerase is doing? RNA polymerase is polymerizing what? RNA based on what? DNA template. So here you guys can see this red part is the RNA, and what is this? Ribosome. This polyribosome will come, attached to this mRNA. Multiple ribosome can come and attach to the mRNA simultaneously, and that's why we call it as this structure as polyribosome, or sometimes we even call it as polysome. So and this translation will happen. So here we guys can see that how RNA polymerase is polymerizing and this RNA, and then how this ribosome is attaching to this mRNA simultaneously, and here the protein synthesis will occur. So here we guys can see that how transcription and translation are coupled in prokaryotes. Now if you talk about the steps of transcription, so first step is initiation. So what happens in this step? In initiation, RNA polymerase bind to promoter region to initiate. It requires sigma factor for initiation. And what is the significance of sigma factor? It is an initiation factor. It basically recognizes the promoter sequence on the DNA, helps in recruiting the RNA polymerase on promoter region. So now I will explain it properly. So here we are going to talk about different steps of transcription. So there are 3 steps. First is initiation, then is elongation, and then is termination. So now what happens in the first step? That is the initiation. So if you guys talk about initiation, so we know what will happen. RNA polymerase will go and bind to what? Bind to promoter region to initiate the transcription. We have studied in a transcription unit, first there is what? Promoter, then there is a structural gene, and then there is a terminator. So RNA polymerase, to initiate the transcription, what will do? It will go and bind to the promoter region. Here is the promoter region. But this RNA polymerase also required a factor which we call sigma factor. What is the significance of this sigma factor? It is the initiation factor. Okay, and it is the one which basically recognizes the promoter sequence on the DNA. So what will happen? This sigma factor, which is the initiation factor, it will go and bind to this RNA polymerase, and this is the one which is going to recognize the promoter sequence on the DNA, and it will, what happen then? This RNA polymerase will go and bind to promoter region to initiate what? To initiate the transcription. So what actually the sigma factor is doing? It is helping the RNA polymerase to get on where? To get on the promoter region. So this is what happens in first step, that is the initiation. Then the second step is what? The second step is elongation. What happens in elongation step? RNA polymerase uses nucleoside triphosphate as what? As substrate. It polymerizes in a template dependent fashion following the principle of complementarity. Elongation of RNA will continues from 3' to 5'. So what actually happen? First initiation will happen, okay, because this RNA polymerase which has this sigma factor, it go and bind to the promoter, and now it has initiated the transcription. Now what is the second step? So the second step will be like this. Now elongation will happen. What will RNA polymerase do? It will use nucleoside triphosphate as what? As a substrate, and even the energy required is also be taken from this nucleoside triphosphate only, because we know that it has what? It has a very high energy bond. But still, let's talk about the elongation step. RNA polymerase will use this nucleoside triphosphate as substrate. Now it will do what? It will going to polymerize in the template dependent fashion, because we know what rule it follows. It follows the principle of complementarity, that purine is going to complement pyrimidine. But here the only difference will be like that A is going to complement with what? U, that is the uracil, not thymine. And G is going to complement the C, that is cytosine only. Elongation of this RNA continues from where to where? It will continues from 3' to 5'. So here we can see this has initiated. Okay, now after initiation, what will happen? This elongation of RNA is happening, and now the last step will be like termination. After the elongation, after the mRNA has been formed, it has to, the transcription has to stop. Now how it will happen? Once the polymerase reaches the terminator region, the nascent RNA falls off. Means once after the elongation step, okay, once now the polymerase reaches the terminator region, okay, now what will happen? The RNA, which the nascent RNA which has been formed, will fall off from there. This also causes the RNA polymerase to detach. This termination process required which factor? Rho factor. So just like the initiation part where the RNA polymerase required the sigma factor, in termination the RNA polymerase is going to require the rho factor. So what will happen once the polymerase will reach the terminator region? So the nascent RNA which formed, means in this RNA we, what will happen? This nascent RNA is going to fall off, as we can see in the picture, that once this terminator has came, this what happened? This RNA fall off. Okay, now at this moment only what will happen? The RNA polymerase is going to detach itself, and it will stop the transcription process. Now this termination process requires a factor which we call rho factor. So this is the 3 step of transcription in prokaryote. First is initiation, then is elongation, then is termination. What happened in the initiation part? The RNA polymerase, which has this sigma factor, okay, it's going to go and bind to the promoter regions. Once it binds to the promoter region, then the elongation part will start. RNA is going to start polymerizing. So after this whole RNA has been formed, then when this terminator region will come, the RNA going to fall off, and DNA polymerase is going to detach itself from there. And it also required a factor what we call termination factor, that is the rho factor. So these are the 3 different steps of transcription. Now this was all about prokaryotic transcription. Let's talk about now eukaryotic transcription. Transcription process is same as prokaryotes with some differences. The transcription process is more or less same as prokaryote, but there are some differences. So what are the differences from those prokaryotic transcription process? Let's talk about that. Transcription and translation takes place at different compartment. Transcription inside nucleus and translation in cytosol, more complex than prokaryotes. So we have studied in this in prokaryote that the transcription and translation are coupled. Means transcription and translation are happening simultaneously, because they don't have any well-defined nucleus. But if we talk about eukaryote transcription and translation take place at different compartment. Transcription occur inside nucleus and translation occur in cytosol. Okay, then more complex than prokaryotes, and definitely the process is more complex than prokaryotes. So, what are the complexity of eukaryotic transcription includes? First is the presence of different RNA polymerases. At least 3 RNA polymerases in the nucleus are present. We have studied in prokaryote that only one RNA polymerase is going to make all the 3, mRNA, tRNA and rRNA. But in eukaryotic, there are at least 3 RNA polymerase in nucleus are present. Different RNA polymerase is going to synthesize different RNA. So here are the 3 types of RNA: RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. So here is the tabular form in which we can understand which RNA polymerase is going to make which RNA. So RNA polymerase 1 is going to transcribe 28S, 18S and 5.8S rRNAs, that is ribosomal RNAs. RNA polymerase 2 is going to transcribe the heterogeneous nuclear RNA, which is what? Which is the precursor of mRNA. Because we know that that precursor of mRNA is going to have what? It is going to have intron and all those things. So this is the going to transcribe the hnRNA, which is the precursor of mRNA. Then RNA polymerase 3, it's going to transcribe tRNA, 5S RNA and small nuclear RNAs. Okay, so these are the different type of RNA polymerase that are going to transcribe different RNA. Now the second point is that processing of primary transcripts, complexity of eukaryotic transcription also include what? Process of primary transcript, because we know that the primary transcript contain both exon as well as intron. Exon is the coding region, whereas intron is the non-coding region. These transcripts are non-functional initially. For that reason, primary transcripts need to be processed. This process includes splicing, capping and tailing. So what we have talked about, that we already know that the primary transcript which got transcribed is what? Has both even intron and exon. But we know that intron is the non-coding part. Okay, and that's why this transcript whole is a non-functional initially. So this primary transcript is need to be like what? To processed. So in this process thing, 3 steps we do. First is splicing, then there is capping and then there is tailing. This figure I will show you, I will make you understand in the upcoming slide when we got to know about all this splicing, capping and tailing. So first is splicing. Splicing involves what? Removal of this non-coding region, that is this intervening region which is introns, followed by joining of exon in a defined order. Means first they will do in this process, what will happen? There will be removal of this intron and then this exon will join in a defined order. Second step is capping. So what is hnRNA? We know that this is the precursor of mRNA only. Addition of methyl guanine triphosphate will occur at 5' end. So here we guys can see. So what happened? First of all, this is hnRNA. This is the precursor of mRNA, and first capping here happened. Capping what happened? At the 5' end of this RNA, methyl guanine triphosphate got attached. You guys can see this GPP got attached. Let's see next. Then addition of 200 to 300 adenylate residues happen at where? At 3' end of hnRNA. This is also what we call polyadenylation. This fully processed hnRNA becomes what? mRNA. mRNA is then transported out of nucleus for what? For translation. So here you guys can see. So what happened? First capping happened, means what happened? This GPP, that is the guanine triphosphate thing, it went and bind to 5' end. Then there will be this RNA splicing where what happened? This intron got removed and then there is joining of exon in a particular order. And then last what happened? This poly addition of 200 to 300 adenylate residue at 3'N, what we call polyadenylation. And after this, what happens? A mature mRNA, a messenger RNA got formed, which we going to be transported out of nucleus in cytosol for what? For translation. So this is the process which is going to happen in eukaryotes. So what was today's class summary? So if we guys gonna talk about today's class summary, so the region of the DNA transcribed to RNA, it contains what? It contains promoter, structural gene and terminator. This is what whole we call transcriptional unit. Promoter is the site where RNA polymerase will bind. Structural gene codes for RNA or protein. Terminator is what? Terminator is the signal which will tell, okay, now it is end of transcription. Now if we talk about the types of RNA, first is mRNA, which is going to carry the genetic code. Then is the tRNA, which is going to transfer the amino acid. And last is the rRNA, which is going to form the ribosomes. If we talk about the transcription process, okay, so there are a lot other types of RNA also. These are the 3 types of RNA that is important in this transcription process. So now if you talk about the transcription process, first is the initiation, in which RNA polymerase bind promoter, which has this sigma factor. Then there is the elongation, in which this RNA strand is going to synthesize. Last is the termination, where this rho factor will work and all this happen and RNA is going to release, fall off. In eukaryotes, there is additional processes in which we have to process this RNA, like intron removal, what we call slicing, then capping and tailing. So this was all in today's class summary. Now we will see some NCERT based questions. So first question says what? That which of the following defines a cistron? Options are a segment of DNA which is coding for tRNA, a segment of DNA which is coding for a rRNA, or a segment of DNA which is coding for a polypeptide. And last, a segment of DNA which is containing the regulatory sequence. So here we know that a segment of DNA which is going to code for polypeptide is what we call cistron. So what is a cistron? So it is essentially a unit of genetic information that is going to code for what? For a single polypeptide chain. tRNA and rRNA gene exist, but definition of cistron is specific for polypeptide coding. Okay, so here what is it is saying that if we can use the word gene when we are talking about that, yeah, gene is something that is going to code for polypeptide chain and tRNA gene or rRNA gene. But if we are talking about cistron, so it is particularly about coding for polypeptide. Regulatory sequence are distinct for coding region, which is the cistron. Means we know that the regulatory sequence such as promoter or terminator, it is not going to code for anything. It is just going to regulate the structural gene. Then second question says, in eukaryotes, structural genes are typically polycistronic, monocistronic, absent, or arranged in operons. So we know that in eukaryotes, structural genes are typically monocistronic. What is a monocistronic gene? So a single mRNA molecules which is going to code for only one polypeptide, one protein, is what we call monocistronic genes. In eukaryotes, most structural genes are arranged in this way only. So this is how a monocistronic gene will look like, that it has only one, it is going to code for only one polypeptide. Whereas in polycistronic, which are present in prokaryotes, there are more than one cistron, and that is going to code for multiple polypeptides. So as I said, that polycistronic genes produce a single mRNA that codes for what? That codes for multiple protein. This is more common where? This is more common in prokaryotes. Structural genes are essential for protein synthesis and are present in all eukaryotes. So structural genes are what? They are essential for protein synthesis, and they are present in where? It is present in all eukaryotes. And if we talk about operons, which we're going to talk more about in upcoming class, is what? A group of genes under coordinated regulation, which is typically found where? Which is typically found in prokaryotes. Next question says, what is the fate of introns in eukaryotic mRNA processing? So, what happened to this intron, this non-coding region in eukaryotic mRNA processing? First, they are translated into proteins. Second, they appear in a mature mRNA form. Third, they are removed by splicing. Or fourth, they are added to the 5' end of the mRNA. So, we know that this answer is going to be third, that they are going to removed by splicing. So in eukaryotic mRNA processing, introns are what? Introns are non-coding regions, while within a pre-mRNA that are removed by which step? Splicing. They are not present in the mature mRNA. So here is, we know that eukaryotic have the split gene, and they have both intron and exon. Intron is the non-coding region, where exon is something that is going to code. So when this precursor of the mRNA going to form, it is going to have introns also. So with the help of splicing, in this step what happened? There is removal of this intron, and this exon is going to add together in a ordered way. So the 5' end of eukaryotic mRNA is capped with modified nucleotide, not an intron. The cap is important for recognition and stability of the mRNA, but it is not an intron. So we have studied that this capping will happen at the 5' end of the eukaryotic mRNA, not on intron, and this cap is responsible for what? For recognition and stability of the mRNA. The next question what says, which enzymes transcribed mRNA precursor in eukaryotes? So which is the mRNA precursor in eukaryote? That is hnRNA, heteronuclear mRNA. Is it RNA polymerase 1, 2, 3 or DNA polymerase? So we know that DNA polymerase is definitely not going to be in transcription. It was required in replication. So fourth option is already out. Let's see from first three option which is correct. The second, that RNA polymerase 2 in eukaryotic cell is specifically responsible for transcribing the precursor mRNA molecule. Whereas RNA polymerase 1, it's going to transcribe ribosomal RNA. We have seen that table also. And the third one is going to transcribe small RNAs like transfer RNAs and 5S ribosomal RNA and all. Whereas we know that this DNA is the enzyme which is involved in what? In replication and not transcription. Next question, capping in eukaryotic mRNA processing involves adding what to the 5' end? So we know that this eukaryotic mRNA processing involves adding capping on the 5' end. So what got added in this 5' end? A polyadenine tail by polyadenylation, an unusual nucleotide, that is the methyl guanosine triphosphate by capping. Third, a ribosome binding site. Or fourth, a polycistronic sequence. So we know that on the 5' end there has been capping with the help of an unusual nucleotide, that is methyl guanosine triphosphate. So it involves what? It involves addition of a modified guanine, that is 7-methyl guanosine triphosphate, to the 5' end of eukaryotic mRNA. This cap protects the mRNA and helps ribosome bind for translation. So this we know. Where is the capping step? This is the capping step where this 7-methyl guanosine triphosphate go and cap at the 5' end. And why it is required? Because it protects the mRNA and helps the ribosomes bind for translation. Whereas this polyadenylation, it occurs at 3' end, not the 5'. And ribosome binding site, it is located within the mRNA sequence, not at 5' end. Whereas if we talk about the polycistronic sequence, the other options, that refers to an mRNA which is coding for multiple proteins, and it is very unrelated to capping. Next question says that the bacterial RNA polymerase require which factor to initiate transcription? Is it rho, is it sigma, is it intron or exon? So we know that intron and exon are what? They are the regions of mRNA, okay? That the coding and non-coding regions. So it will happen that is it rho factor or sigma factor which is going to initiate the transcription? We know that it is going to sigma factor which is going to initiate. This protein subunit is crucial for what? For RNA polymerase to recognize and bind to the promoter region of a gene. It helps in initiating transcription. So this is what this looks like, that this protein subunit is important for this RNA polymerase to recognize and bind to the promoter region of the gene, and that's how transcription begins. If we talk about the rho factor, it is important for what? It is important for termination of transcription. Rho factor does not play a role in initiation. We know that rho factor is needed where? When there will be the termination of the transcription process. Intron and exon, we know that is a specific part of the eukaryotic genes. These are not involved in initiation of transcription. They are related to eukaryotic mRNA splicing. Next question, which RNA polymerase transcribe tRNA? Is it 1, 2, 3 or DNA polymerase? Again, we know that DNA polymerase is going to be in DNA replication only. So in eukaryotes, we have studied that there are 3 distinct RNA polymerase. Each is responsible for transcribing different types of RNA molecule. RNA polymerase 1, it is going to transcribe ribosomal RNA. 2, it is going to transcribe the pre-mRNA, okay, precursor of mRNA, that is hnRNA. Whereas this RNA molecule 3, which is the answer of the question, is going to transcribe what? tRNA along with 5S RNA and certain small nuclear RNA. Whereas we know that DNA polymerase is not involved in transcriptional. Its function is to replicate DNA. Then last question, that which of the following is not a type of RNA found in bacteria? Is it mRNA, tRNA, rRNA or snRNA? So the answer is snRNA. Messenger RNA, bacteria have mRNA which is going to carry its genetic code for protein synthesis. Bacteria have tRNA which is responsible for what? For bringing the correct amino acid to the ribosome during protein translation. This is, okay, I will explain in the next slide. Which is then rRNA, which is ribosomal RNA. Bacteria have rRNA which is a structural component of ribosomes. And snRNA, that is small nuclear RNAs, they are primarily found in eukaryotes only and they are involved in splicing only. So this is how a snRNA look. This is mRNA, this is rRNA and this is tRNA. Now, what are today's keynote? A transcriptional unit contains what? A promoter, structural gene and terminator. Promoter determines RNA polymerase binding, template strand is transcribed. And then there will be like terminating of this transcription process. RNA types, if we talk about it, is mRNA which carries genetic information. Then this is tRNA which is transfer amino acid. Then there will be rRNA which is going to form ribosome. Then there will be transcriptional processes where initiation at promoter will occur. Then elongation creates RNA and then there will be termination which is going to end the transcript formation. So there are a few interesting questions that you guys gonna answer yourself. First, what happens if splicing is disabled in eukaryotic cell? So if there will be no splicing in eukaryotic cell, what will happen? Second, what happens if the sigma factor can't bind to the bacterial promoter? Then why do eukaryote have 3 different RNA polymerases? Then how do capping, tailing and splicing contribute to gene regulation in eukaryote? So every question we have directly or indirectly discussed in today's class. So I hope you guys will be able to answer this. So let's start the DPP of today's class. First question says, which of the following statements about monocistronic and polycistronic gene is correct? First option is monocistronic genes are common in prokaryotes while polycistronic genes are common in eukaryotes. Second says that monocistronic genes code for a single polypeptide whereas polycistronic genes is going to code for multiple polypeptides. Third, Both monocystronic and polycystronic genes are found exclusively in eukaryotes. And last, polycystronic genes contain intron while monocystronic genes do not. So, we know from today's class that what is a monocystronic gene? This gene which is going to code for a single polypeptide, and it's common in which? It is common in eukaryotes. Whereas polycystronic gene is what? Which is going to code for multiple polypeptide, whereas it is present in which organisms? Prokaryotic organisms. So by this, if you will see, the answer will be second, that monocystronic genes code for a single polypeptide, whereas the polycystronic genes code for multiple polypeptides. Save and next. Now, second question says what? In eukaryotic transcription unit, the sequence that are retained in mature RNA after processing are called. So we know that in the eukaryote transcript unit, we have there certain section that we don't want. Okay. But there are sections that are retained in mature RNA after processing, what they are called. So we know that in a precursor RNA, there will be exon and intron. Exon is coding region, whereas intron is non-coding regions. So we have to get rid of intron. So what will be left in the mature RNA? It will be exons only. So answer is exons. Next question, which of the following best describe a cistron? So first, a segment of DNA coding for a polypeptide, a non-coding regulatory sequence, a sequence that is spliced out during RNA processing, or a protein that regulate gene expression. So if we are talking about that, what describes a cistron? So it will be a segment of DNA which is coding for what? A coding for polypeptide. It is actually cistron. It's not citron, it is cistron. Then the presence of introns in eukaryotic genes. The presence of intron in eukaryotic genes complicate the definition of a gene because first, introns are transcribed but not translated. Second, introns are non-coding sequence that do not appear in mature RNA. Third, introns make the gene polycystronic. Or fourth, introns prevent RNA polymerase from binding. So we know that introns are transcribed but not translated. Okay, it is right that introns are transcribed but is not translated, but we are here, the question is asking the presence of introns in eukaryotic genes complicate the definition of gene because these introns are non-coding sequence that is not going to appear in mature RNA. That's why. Then does intron make gene polycystronic? No, it's not like that. Intron prevent RNA polymerase from binding? No, it is not like that. Last question says, regulatory sequence in DNA are sometimes referred to as regulatory genes because first, they code for small regulatory RNA molecules. They influence the expression of structural genes but do not code for protein. Third, they are transcribed into mRNA and translated into protein. Whereas fourth, that they are always located within the coding region of a gene. So we know that the regulatory sequence in the DNA are sometimes refers to as regulatory genes because they influence the expression of the structural genes but do not code for the proteins. So here is today's DBB questions. Thank you. So I'm done, like for today's class I'm done from my side. If you guys have any question, fine, you guys can ask. Otherwise I will end the class because it's already 4:06. Should I end the class? Or you guys have any doubts? Okay. You guys have any doubts? No? Someone say something. Say yes or no, or shall I leave, leave the class, end the class? Okay, then we will meet in tomorrow class. Okay. So go and practice today's class, question, interesting questions, and all those things. Okay, bye.